Hello. Well, welcome everybody. We are waiting for Manel that is almost arriving.
You can wait a bit if you want to.
No, I think we can. So welcome everybody to the first EuChemS Chemical Biology School. So you are starting a new project, a new dream that is to have a European Chemical Biology School, and each year will be devoted to one topic. And for the opening, we chose the Computational Chemical Biology. So welcome, and you are pioneering this experience. And for those of you who don't know what EuChemS is the European Chemical Society. So it's the chemical society that covers all the chemical societies in Europe. And within the chemical societies, we know very well that chemistry is also devoted to study biological problems, and in particular with computational tools. And this is why we are here. All of us are working in computational chemical biology, and well, we wish that this is going to be a very nice time where we are going to learn, we are going to interact, and we are going to get new contacts, new friends, and new scientific ideas. So please let me introduce you, although you already know us. You know Juanjo, Professor Juanjo Nogueira from the University Autónoma of Madrid, and Inés Corral, that is also from the University Autónoma of Madrid, that I don't know if she's able to say hello. Maybe yes.
I think she's connected. Inés, can you say hello?
Yes, I'm here. Hello, everyone.
Okay, so well, maybe she will come for some time these days, but well, she had another commitment, very important commitment. So she's in the background, preparing and helping us. So you are 37 participants from all over the world. So thank you very much for accepting this invitation. Also, we have in total 17 lecturers in total. So you are going to meet many people, and you are going to know many people from mainly from Europe. And well, we hope that this will be an opportunity to learn and to interact. You will need the laptops for all the sessions. I realize that this is not very convenient, but
I was talking to him, maybe we can ask for some tables, because if we have 10 tables like that.
Yeah, there are some also there. I'm going to ask, but because maybe we can have some tables and we can organize them. So
it's not working for people in each table. People work like in each table.
Yeah, yes, okay.
But we will figure out.
Yeah.
Coffee break.
Yes, we are going to try to. And well, before giving you the word, just I would like to tell you that the second edition of the EuChemS Chemical Biology School is already scheduled for next year in Naples, in Italy, and it will be around biomolecular interactions. You know, and especially in our work, how important are measurements and information from NMR, ITC, SPR, different techniques that give us information about biomolecular interactions. This is the chemical school, the chemical biology school for next year. So, well, thank you very much and have fun.
Okay, thank you, Solis. Maybe we can have a look at the program of these days, and then we already introduced Tom for the first lecture. So, just for you to know. So that's the program. It's a very intense program, as you will see. So today we will start with Tom Miclot, who is already here with a molecular. I will maybe use this one here. With the molecular dynamic simulations. So 2 hours of theory, then the coffee break, and then 2 hours of computer exercises. This is why we need the laptops for later. But we will try to use another table, so we will see. Then we have the dinner at 7, from 7 to 8:30. As you know, lunches and dinners and coffee breaks are included. And then after dinner, also here, we're going to have the first round table, and we will talk about graphic design, and EuChemS will tell us how to do nice graphics and plots for our papers and publications and so on. So tomorrow we have machine learning in drug discovery, then free energy calculations after the lunch, and after dinner we have another roundtable where we're going to talk about the scientific career. I will go here because otherwise, for the microphone. And on Thursday we have the theory in the morning, because here we have Wednesday we have theory and computer exercises, theory, computer exercises. But on Thursday is different. We have the theory about machine learning potentials given by Fernanda Duarte, and later coarse grain models given by Manuel Melo, who is already here today. And in the afternoon after lunch, we have the computer exercises related with machine learning potentials and coarse grain. Finally, after the dinner, we have the last roundtable about ethics and risk of artificial intelligence in the simulations. And finally, on Friday, we have the last session about hybrid quantum mechanics, molecular mechanics theory. So before the coffee break, the theory, after the coffee break, the computer exercises, then we have lunch and we go home. That's the plan, okay? So I think, no, we can start. So I would like to, you want to say something before starting?
No, let's go.
Okay. So it's my pleasure to introduce Tom Miclot. Let me check my notes. I forgot already what you told me. So he's expert in molecular dynamic simulation. So he's coming from the Université Paris, right? And he did a PhD at the University of Palermo in collaboration with Florence, right? And then he went to Prague for a postdoc at the Rovsky Institute. And now he is a CNRS researcher at France at the University of Paris. And we are lucky to have you here, and he will talk today for 2 hours about molecular dynamics. Then we have the break, and then the computer exercises. So thank you for coming, Tom, and we are happy to have you here.
Invitation.
I will share your presentation, and we can start. Here is the pointer. So one second. Ines is so you can see the screen, right?
I can.
Perfect. Okay, so the floor is yours, Tom.
Okay, so I'm here and I replace Antonio Mondari, who was not able to come, unfortunately. That is why I make this lecture. And we will talk about molecular dynamic simulations. I hope you will survive. So during this presentation, I will start with the very beginning. So what is molecular modeling? The validity of the classical treatment compared to QM simulations. What is molecular mechanics? Then I will make some 2 words on force field. And of course, I will explain how MD simulation works purely. Then I will explain you how to set up your MD simulations, how to analyze this, and if we have time, some enhanced sampling methods. So, first, what is molecular modeling? It's simply a description of a molecules for which you want to calculate something. It can be CD spectra or whatever. And generally, you describe your molecules, like I represent here, with atom per atoms and using their Cartesian coordinates. So the meaning of molecular modeling is simply to have molecule to translate it into some energy value. Then you want to modify coordinate to lower its energy, and then you perform some analysis. Simple. Now, a little historical point of view. The first methods were QM in the 50s, and they start with ab initio calculations, and then with the powerful of increasing a computer, we can make more type of simulations. And nowadays, and we can make molecular mechanical simulations with obviously the best one, because I use it. And the purpose is to use classical theory to take a look at macromolecular assemblies or dynamics. Nowadays, basically, some people seems to be able to simulate an entire cell. So it's great, but you really need to have big computers and to make a raised brain for that. So basically, it's what I see here. Quantum is more focused on electrons, and the classical method is more focused on 4 atoms coarse grain, or you can make ultra coarse grain to focus on some assemblies. So now the question is, why can we use the classical treatment to describe our MD? The first one is to solve. Normally, if you want to translate a molecule to energy, you must solve the Schrödinger equation. Unfortunately, when you have very, very, very big system like proteins, membrane, or other, it's become very impossible, basically, because you have limitations of really small atoms. So the question is how to solve this. Is the Born-Oppenheimer simulation, Born-Oppenheimer approximation, saying that because of electrons are so small compared to the nuclei, you can make a distinction between the two movements, and basically you can focus only on the movement of nuclei. Then the other is the de Broglie wavelength, meaning that because two atoms, the distance of two atoms are higher than this wavelength, we can neglect the quantum effect between the two atoms. That is why we can go to the molecular MD simulations using a classical treatment. So generally, people already use this molecular mechanics with classical treatment for doping or molecular homology, or when you want to make some optimization of your structure after X-ray or NMR. And you can describe your system in different manner. One can be all atoms, for which you have an energetical landscape very precise, or you can make a group of atoms, and it's called a coarse grain simulation. And in this case, you smooth a little bit your energy landscape. And here, it's basically how coarse grain works. So you have 4 atoms, and you replace some into big sphere. But you will have lecture of this later. So, how molecular mechanics work? and it's not miracle, fortunately. So as I said, atoms are considered as big a sphere. Then between this sphere, you put some strings and you basically shape them. And you compute the empirical energy from the system. So all the terms here are potential energies. And as I showed a little bit here, you try to reproduce the energy landscape. There is good and bad of this type of methods. Obviously, the bad things is that you completely forget for the quantum part due to the simplifications. So you cannot have reactivity or excitative states if you are interested in simply forgot this method. But what is good is that you can study very large complexes like proteins or lipids or some other, and you can see them in a time evolution. So before talking to Andy, we will continue into molecular force field with the base of NV. So what is a force field? For a gamer, it can be things with magic and energy, but unfortunately, it's not video game. So we should here make a distinction between what is in physics and in computational chemistry. In physics, force field is a portion of a space-time in which a constant force is exerted and outside content object of this located on the field. But in our case, what kept our intention is that the force field in computational chemistry is simply what will give us the potential energy surface. So we take all molecules and we compute the energetic empirical potential energy by considering all bonds between atoms plus all unbound atoms. So technically, we take into account the bonds, their angles, torsions, and then we also take into account their van der Waals and electrostatic contact. The first term is to have the energy of a bond. I will explain you a little bit more on this one because it's the simplest, and then we'll go to all the other. Basically, we have two atoms, not matter, who are represented at this sphere, and we have a spring between them. So we have an equilibrium distance, and these two atoms will make some vibrations around this equilibrium distance. And to have their energy potential, we make simply the difference between the distance here and the equilibrium distance. And of course, we add some constants to the equation. So here I give you an example. Here you have the distance between the two atoms. Here you have the equilibrium distance for which the energy is the lowest. And if you increase or decrease the distance, the energy here will change and increase. So if you ask the questions, here it's an harmonic potential, and normally you can also use the Morse functions. But since we are only interested in this acceptable area, both are acceptable. And obviously the harmonic function is the simplest to implement. So here, an example. So remember that we are not in a quantum, we lose all the quantum informations. So we must directly implement properties of the atoms. And here it's a simple example to show with carbon bonds that their equilibrium distance and their constant force should change a little bit. So it means that you need to parameterize your force field considering all these atom types. But I will discuss this later. Basically, here it's the same for angles. We have equilibrium angles, and then they will vibrate around these angles. And using the same meaning, we can recover the potential energy. Here, a little bit in the same way for dihedral angle, but the equations need to have something more with the periodicity of the rotation, with basically here, the multiplicity. So you must use a phase factor or multiplicity, who is the number of minima you have in the function when you rotate the molecule to 30-60 degrees. Then the last one is the improper dihedral angles. So it's the angles when these four atoms become out of the plane. So here, let's say they are really planar, and when one of these atoms move, you have two planes, the original one and the one made by this A, B, C, and then compute the angles here, and this is the improper angle. Then there is some specific case that are not mainly used. They are called cross term. So instead of having, for example, only bonds or only distance, cross term is a mix between two terms. For example, here you have a mix between the distance and the angles. It's very great because you can be more precise in your description of the system. Unfortunately, when you try to parameterize this and recover some constant or equilibrium, it's extremely difficult. And also it drastically increases your calculation times. That is why generally people try to avoid this. But it's simply to know that for you that it exists, and you can have many of possibilities of this cross term. So now, here was all terms for bonded atoms, but as you know, atoms can interact with electrostatic van der Waals. So to do this, we also compute the energy, and we consider this concerns only both atoms that are not bonded. So basically, when you have 1-4 connections, so it's 1 atoms, 2, 3, and 4. No image, sorry. And for this type of interaction, obviously they can happen in intramolecular, so in the same molecule, or intermolecular in a different molecules, when, for example, you have an enzyme and a ligand, for example. And we will see later that we will need a cutoff for this to reduce the computations complexity. So here, a little overview of the interactions. So here in purple, you have all intermolecular interactions of molecule 1 with molecule 2, and in green and in red, you have here all the intramolecular interactions, and in the red here it's all 1-4 connections. So for example, this one, 1, 2, 3, 4, and here they make one interaction. I think there is little problem.
The punto. Is the punto.
Yes, but I cannot switch. So, sorry, so now the lecture is end, so you are free and you can go to the swimming pool.
Yeah.
I can change.
Okay, so so vas a ver.
Okay. Anyway, we have a digital one in case it fails again.
Okay. Okay, sorry for the interruption. So, the first things of non-bonding interactions, and the more simplest, is to compute van der Waals potential using the Lennard-Jones law. So, like before, you use some equilibrium distance, compare them to the distance between the actual distance between the atoms, and then you have the deep of the wall. So here, when you plot these equations here you have the equilibrium distance of the atoms, and here you have the deep of the wall, and obviously these parameters, like all the others, will change with the atom times. The other is the electrostatic interactions. You can compute it with the Coulomb law. It's an attractive or repulsive law because of charges. And it's a very long distance energy. That is why for very big systems, you will need to put a cutoff, because it means that one of your atoms will interact with the atoms very, very far away. And obviously this interactions is not very high. So you need to put a cutoff to save some computation times. One remark, so the effective dielectric constant for the medium is the one for the water or any solvent we want to have. And it's can be very hard to determine, mostly if you are working, I don't know, in some specific solvent. So the solution is to use directly a specific, an explicit solvent. It means that you directly put water molecules into your box with the easiest way to determine it. But of course, by doing this, you drastically increase the size of your box. So you increase the number of interactions and molecular contact you have. So this increases the computation times. And of course, there is some implicit solvent, but there are some limitations. So here, a little quick example to show all of this. We have this small propan 1-pole molecules with only 11 atoms. And from atoms 1 and 2, there is a bond from the 1 and 3. So here we have an angles. For the 1 and 4, here we have a dihedral angles, and we have this one to 3 non-bonding interactions. And then we can continue for the full molecule, but it's not interesting. And here, if we count all the non-bonded interactions, we go to 36. Oops. With only for a small molecules with 11 atoms. So imagine for proteins or protein complex or something with almost bigger, it will become extremely difficult to have this. Then, as I said at the beginning, we lose some informations in because we are using the classical physics, and for this we need to make a very hard work of parametrization for the force field, as it will not work. So technically, the equilibrium distance and angles and all geometric parameters come from X-ray crystal structure of very small molecules. So for example, instead of crystallized proteins, we crystallize only some amino acids or small piece of molecules, and then we parametrize their atom distances, etc. For the force constant, we use the vibrational frequency of molecule that we can have in infrared spectroscopy, and only for partial charge, we need to use QM computations to adjust it. So here, little example, imagine you want to parameterize the bond between 2 atoms. So you can simply download some crystal structure, or if you are lucky, it's already exists in the literature. And then, if you want to have the force constant here, you can make some ER spectrum. You can have it here, and the link between the force constant and the ER spectrum is that you use the vibrational frequency here, exactly, and then the reduced mass of the 2 atoms. But don't worry, generally people already made some parametrization of force field, and generally you can find already all these parameters, else if you are not ready. Then the other things is to create atom types, because here atoms are only sphere, and we know nothing since we lose all the quantum descriptions. So we need to add it manually, if I can say this. And for this, we assign what we call an atom types to different atoms. So for example, here it's the carbon atoms, but depending on its how is it involved in the molecule, so if it's aliphatic or not, you assign an atom types, and then you can change his parameters, so his partial charge, also his van der Waals volumes can change a bit, depending on the situations, and also for the equilibrium distance and angles, etc. So parameterizing a force field needs a lot of work. And due to these parametrizations, they are more or less specialized. So if you parameterize your force field on nucleic acid, it will not completely reach or be usable for proteins, for example. So that is why we develop some general force field, for example, GAP or MMFF, who can be applied to all molecules. Generally, they are for ligands. We have force field for nucleic acids, so one for DNA and one for RNA, because they didn't have same parameters. And it's lab from Olomouc in the Czech Republic who parameterized them. And that is why they are named OL something due to the city of Olomouc. Then we have some force field for proteins like charm, for lipids like bromose, and also for liquids, for basically Ur solvent. And it can be OPLS or TIP3P and some other. Now we know how to have the potential energy, but what we can do only with molecular mechanics is to make some minimization or to simply compute the energy. And in a molecular dynamics, we want them to move. So for different reasons, for example, when we want to see the weightability of a surface, so here it's carbon surface and here it's water. When we want to make, for example, biosensor, so for complex systems, with here it's layer of gold, here I don't remember, and then you have a DNA who is able to target a ligand, or simply if you want to have some information concerning the DNA and a ligand who can be used as drugs or what we got. So, and these simulations start around the 50s with few picoseconds. Fortunately, nowadays we have hypercomputers, so we can reach to microseconds or milliseconds sometimes. And thanks to this, we make some usage in physics, nanoscience, chemistry, and of course, biochemistry to identify how two enzymes, for example, are able to make a complex and to create a reactive pathway in the cell, for example. So, like I say, in biology, proteins or a complex of proteins can adopt different shape, different conformations, and with molecular mechanics, we can only go to the nearest minima here. But our goal is to sample different types of conformations. That is why we use molecular dynamics. So, and this simulation is based on a simple Newton's law. I think everybody already see them, but here a little recorder. And the one who will keep our attention is the second law of the motion, with the force is equal to the mass with the acceleration here. Because, sorry, it's the wrong title here. So when we want to compute a force, like here, we need the mass of the object, its acceleration, and to have these accelerations, we can compute it from their velocity, given the time, and their velocity, we can have it from their vector of position. So here it's the theory. In reality, we didn't use the derivation, but we apply a gradient of the energy. So concretely, what is the relations between the molecular mechanics from which we can have the energy and the molecular dynamics from which we need a force? It's simply this. So the force is equal to the derivation of the potential energy from the position of atoms. But the analytical resolution is not possible, so we need to use different type of approach. And the strategy here is we use some position of atoms, we apply a force field using the molecular mechanics to have the energy here. From the energy, we compute the force, And then I will show you from the fourth, we will compute the accelerations or the velocity, depending on the algorithm. And then we will have the new position of atoms, etc. But this diagram is a little bit weird, so I will explain you later in details. But first, the most important things is the position vector or compass all coordinates in one vector. So all coordinates for all atoms here are represented in one big, very big vector if your system, it's really depends on your system. Then we must use the Taylor expansions here. So I will not explain all these parts, but we cannot directly solve this. That is why we use different type of algorithm. And the first one, the simplest, is the Verlet algorithm. So here you have the procedure, as I explained a little bit before. From structure, we compute the energy using the force field. Then we determine the force. Then we compute the acceleration, and then we can get the new position of atoms. So here, a schematization of the Verlet algorithm. First, you start with your position, then you go to the energy, the force, then you have the accelerations, then you have the previous position, and from this information and this one, for these three informations, you get the new positions. And then you restart, you have the new positions, energy, force, accelerations, you know the previous positions, and then you continue, etc., etc. But unfortunately, this one is not perfect. I will explain why. So first, it's based on the triplet of positions, the last previous positions, and the accelerations of the atoms. And kind of the goal is to predict the future positions of the atoms. But for this, when we start the molecular dynamics, we need the position at, we need the previous positions. But when we start our algorithm, we are starting it here, and we don't know this previous position here. That is why the advantages of this algorithm is it's easy, it's very quick. Basically, you didn't require a lot of data compared to the one I will show you later. Unfortunately, as I said, we miss the positions for the starting simulations. So we need to make some tricks to check, if I can say this. And the bigger problem here is that we lose completely the velocity, the kinetic energy, because the algorithm didn't compute the velocities. That is why it can lead to some error in your MD simulations. And that is why the other algorithms are created. This one is named as a leapfrog, and this one gives us some velocities. It's better. So the protocol is kind of the same. We have the structure from which we compute the energy with force field, then we compute the force, then we compute the velocities from the time step divided by 2, so it's the half time and velocities, if I can say this. And then we can, from these previous velocities, we can have the new positions. So here it's like this. So we start with 4 positions, then the energy, then the force, then previous velocities, then we have the new velocities, then knowing these informations here and positions, we can get a new position, and then we continue like this. So the position at the next step is determined like this. So you know the first positions, then you apply the velocity here, compare with your time step. I will explain the time step later. And then we need some velocities. The problem is remain the same. When we start from here, we don't know velocities, the previous one. So here, this algorithm is a little bit, gives some advantages because we work with velocities, so it gives us a little less wrong in the simulations, but they are not, these velocities are not synchronized, and it requires a lot of data compared to the previous. So here I say that we didn't have the previous velocities. So to cheat, we simply start our MD with random velocities at the beginning. And these velocities must follow the Maxwell-Boltzmann statistical distribution. And because we start with random velocity, your MD simulation need to have some equilibrium phases. So the first times of your MD simulations must be removed, the time for the system to become more physically true, if I can say this. So I hope you survive, because here it's only the beginning. And now we will reach to the setup of MD simulation. So over here, I will start with the overview of the process. There is 2 steps. First, you should prepare your system. For example, if you download some molecular structure, as if it's very small molecules, generally, you need to make some corrections. For example, for proteins, sometimes there is missing atoms or missing residues, missing part of the structure. So you need to have it. And to make these corrections, you can have some other modifications depending on the pH you will run your simulations. So you need to deal with the hydrogens and the pKa, etc. Once you add all these informations, you need to choose a correct force field. Because, as I said, the force fields are kind of specific to a type of system. And if you will use the wrong force field, of course, you will make a wrong descriptions of your system. And then also, if the MD can make some calculations and work, it will not describe the reality on what we will have in experiments. So you will not be able to compare it. Then you should make a solvation. So as I say, you should add the solvent to your simulations and also ions and whatever you want to simulate. And then after all this process, you will have 2 types of informations. One is the Cartesian coordinate of your system, and the other is all parameters in one file. So it's atoms type parameters, atom bond, their bonds parameters, etc. Then, for the MD procedure, you first need to minimize your systems to ensure that the system is in the minimum in the landscape, because you don't really know if it's at the beginning, where is it? Maybe it can be in a maximum and then your simulation will explode. So you need to minimize this. Then, if possible, try to have a global minimization, but with very, very big system like proteins, it can be not possible. Then you hit step by step your system. Then you make some equilibration. And after that, you can produce your simulation. Very easy. So first, the solvation. Why is it important? So basically, we are made in a lot of water or beer, depend of what you are drinking. And it's the basic solvent for all biological systems. And here I show you a simulation of a double-stranded DNA. What will happen if we didn't put any water and any ions? It will completely dissociate. So it didn't, it's not what happened in the reality, as you will die. So then the solvation is also made in different steps. First, you should choose a simulation box type. I will give you some picture later. Then the size of your box, because if you have a big system, of course, you need to change the size. Then you need to add the water molecules, then the ions. For the ions, generally, you can simply add the number of anions or cations you need to compensate the charge of your system and have neutral things. But if you want, you can also put the ions, so cations or anions, in some numbers to have a specific concentrations to reproduce what we can have in experiment, for example. In this way, you can also see what is the effect of salt concentrations of your system. And here, a visual representation of the solvation. You have your system, you choose box type and size, then you add water molecules, then you add ions, and to add ions, you simply remove randomly some water molecules. Then you can deal with really different type of box in the simulations. The most common are cubic, or the truncated octahedrons. So here, generally, you can use it when your system is globular. And what is cool is that this one is closer to a sphere. You have to add less molecules of water, so you can increase your speed up of simulations. Since, for example, compared to a cubic box, you will have less molecule of water inside your box. You can save a little bit of time. But depending on your analysis software, sometimes I didn't really like this type of box. So generally, it's easier to simply use a cubic box. Then, how the box is created. So you choose a distance, and this distance is made from the surface, and you take the maximum distance here, and you have the top, and then you do the same, and you have the bottoms, and then you do the same here for the left and for the right, and then you have your simulation box, meaning that in this case, the distance you choose is the distance between the limit of your box and the maximum of the surface of your system. Of course, to describe water molecules, we need force field. And for this, there is already some specific force field. So the two most used are SPCE and CP3P. And as you can see here, all of them have their own specific parameters and a specificity. So also here, take care on what type of force field you choose to describe your water, because it can make some changes in your simulation. Then, when we have this type of system here, this system is kind of isolated and didn't really reproduce the reality, mainly because we can have some border effect here. So to cheat with this, we introduce what we call periodic boundary conditions. So we reproduce virtually this box in all space in X, Y, and then Z. Yes. And this is to create a pseudo-infinite system to reproduce sort of what you can have in a tube. So here, a little animation to show you what is boundary system. For example, here, if I take this point, it goes through the whole initial cell here. Then we will go to the other virtual boxes here. Technically, virtual boxes are not present in the simulation, and it's simply that we reproduce the when the point here go there, we reproduce it at the opposite of the box with the same direction, same velocity, and the same velocities. Then, to avoid having too much computations, because here, as I said, we create a virtual infinite environment. So when your system is very big at the beginning, you create here a virtual bigger system. And if you try to compute some non-bonding interactions, mainly electrostatics, it will become very, very difficult to have all the interactions possible here. So that is why we use a cutoff. So technically, we choose to make some limitation. For example, here, if I take the example of these molecules, first we try to identify some neighbors of these molecules, and inside this neighbors list, we select only the neighbors that only molecules that are in a certain cutoff, and then all molecules that are in this cutoff, we will make we'll compute their non-bonding interactions with our molecule at the center here, and not for all molecules here. This really reduces the computations time and the memory. Then the other important parameters is the time step, because if you use a time step with too low, your simulations and dynamics will basically not be dynamics and atoms will not move. If it's too big, you will create some collisions between atoms and your simulation will crash. So we need to use a suitable time step. So here I provide you some examples. Basically, if you want to determine a time step, it should be lower than the minimum movement you are interested in. So if it's for flexible molecules and for atoms, generally it's this 1 femtoseconds, because we take in account the vibration of hydrogens. If it's flexible molecule, rigid molecules, we can increase it by 2 femtoseconds. And then if it's, for example, with ultra-coarse grain, you can increase this this time step with 10 femtoseconds. As I said, in MD simulations, the time step is around 1 femtosecond with slow, and we need to increase the computation speed. So for this, we cheat. Sorry, there is no word for this. We use some trick, and it's the shake procedure or the settle procedure. The first one add a force to covalent bond with hydrogens to make them rigid, and the other, the settle one, fix the geometry of water molecules. So the solvent is become rigid, and thanks to this, we can reach from 1 femtosecond to 2 femtoseconds, meaning that between 2 steps of MD simulations, you have 2 femtoseconds. But we can do better using hydrogen mass repartitioning. So here it's very simple. We artificially increase the mass of hydrogens of our systems. Will become from 1 to 3, and because we need to an equilibrium in our system, all atoms that are bonds to these hydrogens will see their mass reduce to have some compensations. And thanks to this, we can reach from 2 femtoseconds to 4 femtoseconds with very great. But take care, because first, it's not applied to the solvent. And if you do this, because we change the mass of hydrogens and make some other changes, depending on the analysis you want to do, you may be not interested in all these accelerations process. So, for example, if you want to make some QMMM, it's better to didn't take them in account. If you want to correctly minimize your system also, or if, and of course, if you are interested in hydrogen vibrations, since we keep them rigid and change the mass, you can completely forget about it. So depending on what you are interested in, you may want or not use this type of process. Then we go, so we make, we correct the structure, we make the solvations, and now we need to ensure that our system will not explode. So for this, we'll make a minimization. So generally, the global minima is never reached, mainly when you have a very, very big system like proteins or complex of proteins or whatever. And we go to the nearest minima here, simply to ensure that the initial energies and velocities will not make our system explode. And here also in this step, the time step you choose is also very, very important, because here we will make atoms move a little bit to ensure that the structure will converge to the minima. And for this, if your time step is too short, you will never go to this minima. If it's too big, you will simply never converge to any minima. So you need to choose the good one to ensure that you go where you want to go to this minimum. Then there is other informations you need to know, is called statistical set. So basically it's some thermodynamic constraint on your simulation spots. You have different parameters. So there is the number of atoms, pressure, temperature, the global energy of the systems, and the volumes of your box. And there some statistical set exists. And for this, we fix some of these parameters. So of course, the number of atoms is always fixed because one atom will not magically appear or disappear in the simulation. There is the MBE environment, statistical set, sorry, for which we fix the volume of the box and the energy. So it means that we let the pressure and the temperature have some variabilities. Then it's the MPT, for which we fix the pressure and the temperature. It means that here also the volume of the box can change and as well as the global energy of the system. And the last one is the NVT, for which we fix the volume of the box and the temperature, and we let the pressure and the global energy have some variability along the NV trajectory. And obviously, if you want to reproduce experimental conditions, it's better to use the NPT. But sometimes, for some kind of use, you may be interested in some of these. So now I say that we can keep some variable constant, like temperature on the or the pressure. And for this, we needed to make some thermostat or to apply a barostat. So the first thing is that all MD simulation can be described as classical Hamiltonian with the kinetic energy and the internal energy. So this one is computed with the force field. And the kinetic energy can be recovered using the velocities. That is why it's important to have and to know velocities here. And then if we scale the velocities, we can maintain the kinetic energy and of course the temperature constant, because we can link the kinetic energy here with the temperature constant using this formula. And we use a scaling factor here all along the simulations. And thanks to this factor, we can keep a target temperature. Then, if we want to keep the pressure constant, we need to use a barostat. And this one, we use imperial theorem of Clausius with the equations here. And basically, the barostat is the sum of ideal gas part with the contribution to the particle interactions here. Basically, to keep 1 atmosphere pressure, it's easy for isotropic system, but if your system is anisotropic, just try to solve this, because you can encounter some problems since it can become very complicated. So then I go to the really last part, I promise. It's to correctly describe the electrostatic interactions. Again, I'm sorry. So basically, some diapositive before, I say that we can simply use a cutoff with through and it works. But if we do this, when we use a simple cutoff like this, we have some artifact in the energy descriptions, and it makes some wrongs because of the border effect of the cutoff. See, and the strategy is to use the eval summations, as you can see here, who describe a little bit better the evolution of the energy. So it's these very long equations here. It corrects the error of the cutoff, but it's very, very, very long to solve. That is why we use a simple strategy. Instead of solving directly these equations, we use a fast Fourier transform to speed up the eval summations. And this strategy needed to work in a discrete data. So we cannot take the entire box, but we divide this box into a grid, and we make a repartition of charge into this grid. Then we compute the eval summations with Fourier transform, and then the new charge positions is set on the simulation. So I show you how to set up your MD simulations, and now it's time to analyze it, because it's cool to have the simulations for trajectories, but it's also good to have some information from it. And the first analysis you must do is to check that the parameters of the MDs are coherent. So it's not directly your system, but it's more, for example, here, the total energy of your system to ensure that there is no some problems in your simulations, because it will be roughly stable. And if you see your energy moving like this, maybe something wrong in your simulations happen. Unfortunately, this type of problems can occur since your trajectory is running without any error message, and you can see something moving. But if you see some too much variation in your energy, it means that something wrong may happen in your simulations, and you need to investigate this. So it's true for the total energy, but it's also true for all independent energy. I mean the potential energy or the kinetic energy, who also must be stable. And of course, you also need to check other parameters, so the temperature, the pressure, the volume of the box, and see if it's coherent or not. For example, if you set the volume of the box in your simulation as constant, and you see that this volume change also can happen, and there is a problem somewhere. Maybe it's because you choose some wrong parameters, or because of some algorithm problems. This is important to check. Then you also need to check properly your system. And for this, you have different type of strategy. So the simplest one is the RMSD. So it's the quadratic distance between a structure of reference and the structure at a given time. Generally, the structure of reference is the structure you have at the beginning of your simulation, but it can be some other if you want. And then, of course, so you compare here the position of atoms of your structure at a given time compared to the dispositions at the beginning of your simulations, or any reference you want to compare your trajectories. And if you have a value of 0, it means that there is a complete superpositions of these two position vector. So basically you have exactly the same structure. Here, I give you some examples of RMSD on the simulations. So if the RMSD of your simulations completely increase or is not constant, it means that your simulations didn't make any convergence. So for this, you have two solutions. Or you simply continue your simulations and wait it for some updates and convergence. And if it's not possible, so you can try to restart your simulation. Maybe it's because of the initial trajectories that are not set correctly. It can happen, or it can be an error in your system. For example, if your system are too close or whatever, it can be an error also in the systems. Generally, not really in the setup of your simulations, let's say. So a correct RMSD should be like this, so globally stable. We see that at the beginning there is an increasing part. So it's when your simulations try to become in the equilibrium, and then when the RMSD reach plate here, it's when your simulations become stable. And what is cool with RMSD is, for example, here, without any visualizations, we can see that there is some movement or some structural change in the simulations. And thanks to this, you can have some quantifications between the structure changes. The other is similar to the RMSD, but it's the RMSF. And instead of using the initial structure or any reference structure, we use the mean here, the mean positions. So it's basically how one atoms or a group of atoms, or what you are selecting, fluctuates along the simulations compared to their mean. And if you make some X-ray experiment, or if colleagues do this, you know the B factor with kind of similar, and it's possible to make the correlations between the RMSF and the B factor using this formula here. And here, I give you the comparisons between the B factor here in blue and the recovered B factor using MD simulations here in orange. So it's pretty similar, and it means that we can have also this type of fluctuation information. Then you can have other parameters you can be interested in. So one is the solvent accessible surface. So it's this. We have a ball who will roll all around your molecules, and this will give you the surface of the surface of contact of your molecules with the solvent. Here it's very interesting because, for example, when you have some changes or associations, the surface accessible, the SASA cannot change, and this gives you some hydrations informations of your proteins or what you are studying. Then it can be interesting in the gyration radius. So this radius gives you informations about the compactness of your system. So I will give you an example. Imagine you have two systems. One is globular, so it can be a sphere, and the other is a cylinder. Both of these geometric shape can have the same volumes, but their compactness is different, since a ball is very compact and a cylinder is more longer, so it's less compact. So this gyration radius can also give you some informations. For example, if you are interested in some folding or some aggregations things, it's very interesting to compute it. Then we can have some other information, so the densities. For example, you can divide your simulation box into a big grid, and on this grid you can compute the number of atoms and see the repartitions and of your atoms into this grid. For example, if you are interested in the we don't know, in the repartition of ions in the simulations. For example, you have charged molecules and you want to see how the salt will aggregate or not, you can use this number of atoms densities to determine if the ions will converge or not to your molecules. But you have different type of density. Obviously, you can have the charge densities, the mass densities, polar densities, and whatever you want to make as densities. Also, you can make some specific analysis with not directly correlated to the physics or to the chemistry, but more to the biology, I want to say. So it's the interactions between amino acids or nucleides, if you are interested in DNA. And for this, you can track the evolutions or, for example, charge or repulsions or clashes. You can, of course, the classical hydrogens bond or van der Waals contact, but you can be more specific. For example, chalcogen bond, so the interaction with sulfur here with carbon here, the NP interactions, amino P interactions, and whatever interactions you are looking for. There are many. And that is why I make a little publicity of my software. So it's a Python software, Molecular Interaction Analysis Toolkit, or Miclot, like Tom Miclot, by Narcissism. Little joke. So this type of software, I implement non-bonding interactions like this, but if I make the declinations all possible, you can have an overview of around 30 interaction types. You can also put your own parameters. For example, if you are interested in some PP stacking and want to use some parameters of angles or distance, you can put it directly. It's not strict like the other software. And of course, I also implement some cysteine bridge, because cysteine can have a selenium atoms. So two cysteine can make disulfide bond. So it's a bond between two sulfur, or diselenium bonds, or selenosulfide bonds. So it's bonds between the selenium and the sulfur. I don't know why it's not very you didn't have many information of this type of bond in the literature. They exist, but people are not very interested in, however. And of course, you can also easily have some structural information. So for example, if there is some flexible loop who can, due to some external interaction, can become an helix or other, you can also follow this. And some other interaction. Then it's also interesting to have some visualizations, of course, to have very good images to put in your paper, but also to visualize and try to understand a little bit more structurally what happened in your MD simulation. For example, if you see your DNA completely dissociate, like I showed before, or here to show the interactions between the DNA and the molecules. It's interesting to see your trajectory. And for this, you can use DMD or the new version of Chimera. They call it Chimera X. And of course, this type of software can make some visualization, but also some analysis like RMSD, RMSF, and some others. So they are very interesting. Then, okay, perfect. I'm in time. I will give you a very overview of enhancing sampling methods, but I saw in the program that you will have some free energy lectures. So normally you will see it in really more details. Here I discuss about equilibrium MD simulations with good methods. So you have the evolution of your system with the time. but the time we can reach is generally microsecond, and using some tricks you can reach to microsecond. For example, if you use Corey's grain. Unfortunately, in biology mainly, some system can make some structural change or some conformational change or whatever in time with more than microsecond. So using this method, you simply cannot see it, unfortunately. So the strategy will be to replace the time description by an energetic descriptions. So it's basically what I say here. And this strategy, you induce a potential into your simulations to ensure that they will reach some barrier. So for example, I make an illustration here with the meta dynamics. Here, let's say it's the result of our MD simulations. We have the system with here, but what we want to see is not here, it's here. So basically using this equilibration time MD, it's not possible to go to this barrier and go to the other conformations. So the strategy is to put a Gaussian here, Gaussian potential, and at it little bit by little bit. Thanks to this, we will push our system across the barrier, and it will lead to the other minima here. So using this type of strategy, we can see different conformations of our system, and obviously we can accelerate the time. For example, if you are interested in some aggregations, it can be very useful for you. Then there is also some problem with accuracy. So you need to choose a very good definition of the weight and of the width of your Gaussian, else your system will do nothing, basically. And also you need to choose a correct collective of variables. So a collective of variables, it's whatever. For example, it can be a distance. If you want to put a ligand close to the catalytic space of the enzyme, you can put it and stride it to positions. You can also use RMSD. For example, if you have the crystallography of two conformation of the proteins, and you want your proteins to go from one to the other, you can use this type of collective variables. And it's really, really not easy to choose. Nowadays, people try to use artificial intelligence to help people to choose this type of collective variables, because sometimes you can choose one collective variables, you think it will work and it will not. Then you can choose, you can also combine a different type of collective variables, and it can work or it can not work. I have an example in a publication I made later. It was an enzyme able to cut DNA. So fortunately, we see a paper who use some an equivalent system. So we tried their collective variables, and it didn't work for our own system. Also, if the enzyme and the global things look the same, and we have to choose some other collective variables, and we play with many of them to try to make the reactivity of this enzyme. So then a little bonus with here the personage from Accelerates. I will give you some good references here. So it's mainly books. You can read them. And if you are mostly interested in enhanced sampling, you have this review made by Jérôme Hénin and Lucie Delmotte. and you have, in a simple way, all the many of the techniques you can use for the enhanced sampling. So I thank you, and now we have some time for the questions. If you have questions or not.
These slides can be sent to us. And the presentation will be
This one?
sent to us.
I don't know.
If you agree, yes, we can do it.
Yes, I agree, so you can have it.
I asked your director if they agree, I will send you the slides.
Or if you want to take photography, I can also.
You can go one by one. Yes. So the stuff.
Okay, so now it's time, if you want, we have some time before the coffee break for questions for
Yes,
Tom
so I
regarding
plan.
the lecture.
Yes.
I have a question about when you want to parameterize, for instance, your ligand,
Yes.
what things do you have to take into account? I mean, I have seen different protocols, different levels of theory,
Yes.
and sometimes it seems that everyone uses a different method or they have their own ideas. Because
Yes.
what I try to do is to imagine I have chosen a force field. For instance, if I have chosen a force field,
Yes.
I look at the literature about that force field and see how it has been parameterized. For instance, the QM that I have used for computing the charges and all that stuff. And then I would use the same level of theory to compute, for instance, the charges for my ligand. But I am asking this question basically because I use Amber, and I have seen in different forums that they use Hartree fork to compute the charges.
Yes.
Because in principle, with Hartree fork, there is some overestimation of the charge, which can reproduce somehow the solvent effects, things like that.
Yes.
But in some situations, I have been questioned about this. So I always struggle to do to choose between one level of theory and another. And regarding also the same thing, when you want to parameterize, you start from a random conformation from the ligand. Imagine that. But that conformation, once you dock, for instance, your ligand in the enzyme, it can change quite a lot. And in some protocols, the ligands are optimized, and then the charges are computed from that optimized structure. If your structure is optimized, it isn't actually how it is the conformation in a static picture in your enzyme. Well, the question is related about all that stuff, basically.
So it's basically true, but it's depend on how you want to parameterize your ligand. So you can go to the simplest way, and for example, use the gap and the compute some molecular charge. In this way, it's not necessary to have the optimized structure of your ligand. And generally, the gap have the general parameters, so it's the simplest way to do. If your ligand have metals or other, it's you cannot use it, unfortunately. So in this way, you need to read what is in the literature or to parameterize yourself. I know that Amber create a script for this. If your ligand have some metals, I don't remember the name. It's MCPV, who is very great. You can do it basically everything automatically. And yes, sort of. It's easier than if you do it everything manually. So it really depends on your ligand and on your system. There is no perfect answer, sorry. If it's purely organic, the simplest way is simply to use the gap and think I make, I will show you. There is a GUI for parameters. If you want, you can use also Charm GUI, who can help for parameterizing ligand, mainly if they are organic, purely. So you can try this. But if you have specific things in mind, it can become very difficult. So there is different possibilities.
I have another one. The other one is about the initial structure of your enzyme, because I am doing molecular dynamics, usually with an enzyme and a substrate, like a complex, and I always struggle to decide whether it's better to, once you have a structure, run apo-molecular dynamics without ligand, and then dock your ligand into the structure, or start with a static picture that comes from whatever, with resolution or alcohol, or I don't know, what do you recommend?
So if you already have the structure of the proteins with the ligand, it can help, so you can do it. But generally, what I do is I make multiple systems. So it's the ligand alone, the protein alone, the complex of the protein and the ligand, if it exists. Else, you can try some docking. I'm not sure about AlphaFold. I'm not sure about the it's really very precise or not. And for what I see in their website, for AlphaFold 3, it's not really mandatory to use their structure to make MD docking or other. So you can try. And if you didn't have the structure of your protein and the ligand, you can use accelerated MD simulations, or you can simply try different docking poses. For example, if you use ADOC, from the best docking part, you have several structures, and you can simply try different type of position. Or you can make some steered MD simulations also.
Okay, thank you.
Let me add something to this question, because we have a paper where we run MD and then we do docking on top. And if you have the ligand protein system, and ideally you should take this structure because the active site is already open. But if this is not the case, what we do sometimes is post-solvent molecular dynamic simulations, where we do mix water and a non-polar solvent with water, because in this way you open the non-polar properties also. Like you can do water benzene 80–20% or something like that. And this is a very efficient technique to open the non-polar properties. If there is no structure where the ligand is already there, that's very useful. This is called post-solvent.
Thank you.
My question is for the what do you consider for the solution of the collective variables? For example,
It's really depend on your system. Yeah, unfortunately, there is no specific protocol to choose collective variables. That is why people try to make some artificial intelligence on it to help them to choose correct collective variables. The classical one is, and the simplest is to play with distances. So you can play with the distances of atoms or group of atoms. And then it's really system dependent. So I can really not answer precisely. But if you want, I think it's in the Amber, no, in the new versions, it's the group of Iñaki. They make a method from which you can put multiple collective variables, and then the software will play with all these parameters and then choose the right one to make whatever you want with these collective variables. Yes. So now it's implemented in Amber, and the group it's Iñaki from Iñaki. No. Yes, in Valencia.
Thank you. Thank you so much.
You're welcome.
Thanks for the presentation. For maybe those of us who haven't done any MD calculations before, can you comment on the different types of software packages that you can use to run these simulations, and sort of is the procedure roughly the same for all of them?
Yes, basically procedure. So here and here, the overview it's the same. It's simply the tools you use. So for Amber, you use a TD. Gromax have its own tools. OpenMM also, and basically the overall is the same. It's more the implementations on how calculations are done, or is a difference. For example, if there are more CPU or GPU, and also it's the methods that are implemented. So, for example, I know that in Amber you have different type of methods you can use to make different type of MD simulations or enhanced sampling. Sometimes, for example, the Gaussian accelerated MD is implemented in Amber completely, but it's partially implemented in OpenMM. In OpenMM, you can only play with some non-bounded parameters, not all parameters. And I didn't know that if it Gromax, it's implemented. So it's really depend on your affinity of the software first, and the method you want to use. But basically, you can reach to the same things.
Yes, it's kind of like the analogy for stuff where I like the gas you go for them all very almost like it, and there's genes that can go the same gas benefit.
Esta es una cuestión Miclot.
My software, yes.
Does it use a particular trajectory format?
No, so it's based on MD trash. So it's what MD trash can read, he can read it. And for some part, I use OpenMM, but for this, I convert the MD trash to OpenMM. So you can have read trajectory from Gromax, from Amber, and what MD trash is able to read. For you can also read a PDB file, a simple PDB file to prefer.
Okay.
I would like to ask to add some comments regarding your question. You were asking about the parameterization of the ligand, but this is absolutely crucial. And partial charges, and if the parameters are reproducing the conformation of your ligand, for instance, in carbohydrates, this is a very important question. So maybe you have to devote time to parameterize your ligand, to perform simulations of the ligand in the free state and analyze the conformations to check that all the parameters are reproducing the behavior of that ligand. So, well, I just wanted to make this clear because sometimes we need to devote a lot of time to that, but it's absolutely important. Okay, so and sometimes it's a lot of work, but we have to do it. Okay.
There is a question from somebody in YouTube, and he says, how do you solve the issue of equilibration if the root mean square displacement is deviating too much. It's fluctuating still too much.
Good question. So this it can mean that, of course, your system didn't reach to the equilibrium. So maybe you can start to make a longer minimizations. Maybe it can happen because your initial system is not very well minimized. It can happen. Else, did you try to run some other replicates? Because sometimes I know that you can have some bugs in the MD code, and you can have this type of error. For example, it's not directly due to the system or the algorithm, but if you have some error in a GPUs, if you use a GPU, sometimes you can encounter this. Can happen in the cluster. Else, if it's really your system, think it can be. I have no information about the system, so it's difficult to answer. But for example, if you make some manual docking because your two proteins are too big, so you try to put them together. Since it's manual things, of course, you can have this type of problem. Else, yeah, it's all I can say since I didn't really know the system.
Okay.
Other questions?
Sorry?
Other questions from the.
That's the only one so far.
Okay.
More questions here?
Yeah, thanks for your part. Based on your slide for the MD production, MDT or MDT, which one is better for launching the MD production?
You mean
For the step 5,
this?
because MDT is more similar to the experimental
Yes.
condition, but in lots of papers, the people they are launching MD simulation with MDT. Which one do you think is the best, or it will really affect the simulation or not?
Generally, NPT, it's better since you can easier deal with experiments, so you can easily compare with it. And for NVT, it's only matter of the pressure. So if you track the pressure and you see, well, you will see some variation, but normally it didn't should differ so much. So if you have very, very big variation, there is a problem, but generally it didn't need to change so much, and we have some stabilization. So in this case, if you simulate simple proteins or things, it doesn't really matter. It's more when you want to make the equilibration part. Sometimes you can play with all of this to ensure that your system will not explode. Who can be why the RMSD and become so high, and your system didn't converge because the minimization or the equilibrations at the beginning was not done properly.
And the other question is related to using the constraint during the equilibration.
Yes.
If by applying the constraint during the equilibration, you can some kind of abandon the jumping in the RMSD by using the constraint just to avoiding some artifactual movement of the ligand, or you don't suggest to using constraint?
So generally, when you have some flexibility, for example, with ligands, can be very small and walk around. the best way is to make a multiple state equilibration. So first you apply a bigger constraint in all your system. Then, for example, if you have two proteins or two chain and a ligand, you can slightly decrease one chain, then the other chain, and then the ligand, and go really step by step, and by decreasing the constraints in the whole system or part of the system to ensure that your system will relax, if I can say this, slowly. Sometimes it's important, as you say, mainly with small ligand molecules, they can simply go.
So there is time for one last question. Okay, if this is not the case, I will check the one YouTube the last minute. No, this is not the case. Okay, so then let's take Tom for the talk and for the lab. Okay, so now we have the coffee break. We go back to the main building where you register when you arrive, and then we are back to 5 o'clock. Half an hour. Okay. Thank you. So we will, at the end, we have tables enough, so people working there, we will reorganize the team because we have 10 or 11. So 4 people in each table, it comes.
Because here
so you can go to the coffee break and start. We are trying, Martin and they are trying GOMAX in the supercomputer center. Kind of is working well, so but later they will tell you how to log in or
I take my
for the no, it's better use. You have everyone, right? Wi-Fi.
I don't have my selection.
If not, we can use mine because I have okay, or maybe here there is here we have a Wi-Fi, you know, I guess Wi-Fi. And if not, we can ask people to study because it's not here. Stop.